right. Hello. Let's go. Back at it. Actually, let's make me a little smaller or move me a little over. There we go. There we go. Okay. Yeah. Let's talk SARS, coronavirus 2, and intracellular immunity. And there's a lot to talk about here. So let's go back to the beginning. We're going to cover disease first, and then we're going to cover how the virus is actually able to do this by basically evading the intracellular immune response. Okay. So, step one, what does coronavirus disease look like? Coronavirus typically causes two different kinds of disease. One is a respiratory disease, which is going to be like what you see here, and like what you get in MERS and SARS and SARS-CoV-2, COVID. The other one is going to be an enteric disease, which would be diarrhea in a word. And then you got various exotic coronaviruses that do all kinds of other horrible, lethal, weird things. But the general trends are that those are the two most common things, respiratory and enteric. Yeah. And so a respiratory infection would look like this. So you would have, this is actually a fairly bad case. What you want the lungs to look like is sort of the top of this lung. You want it to be nice and just sort of black appearance as you're looking at it. But what you see with infection is you get all this sort of infilling. You get a lot of scar tissue, you get edema, which is like fluid building up in some of these spaces. And it's just a whole bunch of stuff that makes the lung unusable as a lung. And so that is something that is very bad about the virus, but that's not always the thing that kills you with the virus. There is another more insidious mechanism. So let's talk about it. Okay, so when the virus comes into a cell, we know it's going to need to bind to ACE2 and to one of the proteases. So TMPRSS2 is going to be the most common one on lung cells. It can also go in by a different route. There's a thing called a cathepsin enzyme that can cut at the same site, but it does it in a different way compared to TMPRSS2. Anyway, as the virus grows, it will set off the cell, and the cell will start to release basically some signals that indicate that it is infected. Now, some of these signals will go to nearby cells, such as this guy, this green little pirate hat shaped dude down here, which is the alveolar macrophage. So this is an immune cell that is like embedded right there in the lung. It's just patrolling, that's its beat, and yeah, waiting to see if anything bad happens. And so that alveolar macrophage will then get activated. And when it gets activated, it will send out a whole bunch of signals. So these will include things like MIP1 alpha and MIP1 beta, interleukin number 6, MCP, and IP10. These are highly pro-inflammatory cytokines. And so these are basically a call for help. These are a call for other parts of the immune system to come in and do something. They are part of a signal. So normally the cell that is infected would make a thing called interferon, and that would identify that cell as the one with the problem. And then the macrophage is here to amplify this distress call and bring in lots of help in the form of other immune cells that will then seek out the cells that are making interferon and will destroy them as a way to destroy the virus. They'll just sort of root out anything that's in that area and just, wipe out all the cells in the area. It's very thorough and maybe not what you would want, but it's the way it works, and generally we can survive this. Okay, so when you've got an alveolar cell and it gets infected, it's going to have a macrophage. The macrophage is going to send out some signals, etc. Now, one of two things can happen there. Either the body can figure it out, can make a very good immune response with dendritic cells and all these other cells from the immune system that you'll totally cover in immunology someday. In which case, it's going to be very specific and it's going to shut down virus growth. Or you can end up with a lot of undirected cells coming in. So you get basically very riled up macrophages, And they call in these other cells that then release a bunch of chemicals. And basically, none of these cells knows quite where to attack because the virus is masking its presence. If the virus is able to hide pretty well, then then the immune response becomes much less effective. And the thing is, with an ineffective immune response, if you have macrophages and they are not able to find the thing they need to kill, they'll call in more macrophages that'll also release all the same chemicals. And you get one of these, it's called an uncontrolled exuberant immune response. And it would not make you feel exuberant. This is basically the same mechanism that causes disease in Ebola. Same mechanism that causes disease in bird flu, yeah. And in COVID and in MERS and in Hendra and Nipah, a whole bunch of other bad viruses. So this dysfunctional immune response with lots of macrophages and monocytes just releasing all their contents, that is the bad thing. That is the thing that causes blood clotting and causes destruction of the heart muscle and causes damage to the brain and causes just real severe damage to all the blood vessels. And that's what you don't want. That's what is associated with hospitalization and or death from severe COVID-19. What you want is a nice specific response that gets made really fast, shuts down all this macrophage nonsense, and basically comes in. Because the macrophages will be quiet and go away when the right T cells come in and say, it's all right, stand down, we've got this. Question is, do you make the right ones in time? Do you get them to the right place? And that is literally the definition and the difference between life and death. It's whether you have a good helper and killer T cell response and all the things that go with that. All right, so that was our little disease corner. And in the next segment, we'll cover interferon, what it can do, what it can't do, what it should do, and what it doesn't do. Yeah, stay tuned.